Assalamu alaikum. Good morning. It gives me an immense pleasure to start this first session of today. Today is the second day of the conference, and we are honored to have Professor Vittorio Calabresi from Italy. In Italy, we say Buonasera for good morning. And he is a well-renowned professor from University of Catania in southern Italy with more than 100 publications, several patents and books to his credit, and he has done a lot of work on Boswellia extracts, and today he will tell about its effectiveness in traumatic brain injury. Those of you who are not familiar with Italy, Italy has a long tradition of medical schools dating back to 800 years. Two universities in northern Italy, Bologna and Padova, which had medical schools 800 years back, and they have been continuously running all these 800 years. Welcome, Doctor.
Thank you, Mr. Chairman. Good morning, everybody. And it is a great pleasure to be here sharing this interesting time with all of you, and a special thanks to Professor Rashan, Professor Amar. They have been so kind to invite me to share with you the object of my present enthusiasm. And there's something in automatic that goes, so I will be fighting with time, but this is a good challenge. So in a few words, if I want to really give a message of what I'm gonna show you about my interest is automatic nutrition, these few words, automatic nutrition in neuroprotection, from bench to clinic. I want to exploit this possibility to convince you that it's a good table for possible interactions. And everybody is aware of the increasing lifespan, which means a lot of depth with evolution, because we know how to live more, but we don't know how to live better. Yes. With the increasing lifespan, of course, the aging issue is at the frontier of science interest. Just as a brief overview, this is the rationale that has been considered to create this new area, is anti-aging medicine. Actually, I'm the vice president of the European Academy of Anti-Aging Medicine, which means also doctors that are anti-aging doctors. It's really a new area of medical investment. And all started in 2004, when I was in New York. It was a very fortunate day when I discovered the role of curcumin in A-beta, preventing A-beta deposition. This had a very worldwide resonance. All journals, they reported this fruit of collaboration with New York Medical College. And we are very familiar with the role of polyphenols, like curcumin, but also a number of other polyphenols, the way they activate so-called vita genes. I've put in motion this idea of vita genes. They are genes that are redox sensitive, that potentiate cytoprotection. These vita genes, they are centered around NRF2. Now, what is fascinating, it's just a matter of months, is emerging a double role for NRF2, canonical and not canonical. So we will see canonical NRF2 activation through KEAP1, but also non-canonical through P62, which means transient activation, which is good, or persistent activation, which is not necessarily good. And this is a new frontier of NRF2 world. And my Vita genes fundamentally recognized with especially its shock factor and its shock protein, NRF2 regulated genes like heme-oxygenase, gamma-glutamyl cysteanyl synthase, which is crucial for glutathione, C2 thioredoxin system. And what is neurodegeneration? Just a few words and then we go with data. What is neurodegeneration if not a problem of apoptosis? There cannot be any Nobel Prize discovering how to cure Alzheimer's, Parkinson, multiple sclerosis, or neurodegeneration without thinking to intercept the way how neurons die to prevent it, which is apoptosis. And what is cancer if not a problem of apoptosis? I cannot imagine to win against cancer without going through facilitating the way how a cell can die, through apoptosis. So this is a very old luck that maybe we can have a new key. The same key from the way I rotate can control, who knows, but we cannot say nothing in science if we don't measure. Here, for instance, we can see how many sites, for instance, apoptosis is prevented by upregulating its shock protein. Here, here. This is upstream of the canonical apoptotic pathway. So if I induce heat shock, I prevent apoptosis. But where? In normal cells. Post-mitotic cells, like neurons. Not in cancer, perhaps, which is the case. So let's consider heme-oxygenase as an example of beta genes, NRF2 regulated. If I through microarray transduct the promoter of human heme-oxygenase, this is the results. Apoptosis, upregulation of anti-apoptotic genes, BCL2, a downregulation of BAX for apoptosis. So the signal for apoptosis is prevented. And this paradigm has also the other side face of the issue when in cancer cells, if I induce heat shock signal, I facilitate apoptosis. Why? Because heat shock synthesis is a high energy demanding process. It goes in conflict with hyperproliferation. So the system goes phase and then apoptosis. And this is a matter of publication in Nature. And this is fostering my motivation, go deep in the role of neurogasobiology in neuroprotection. And coming back to the NRF2 sensitive genes and the way how I can impact the redox system is a way how I can answer to a question. So far we have added here to life. How I can add life to here? I mean, I can have as a clinician, anti-aging doctor, I can have two centenarians, two. One come to me after running, after in good shape, another in a wheelchair. I don't know if he will die in 1 week. They both have 100 years. So the way how I can contribute to make the quality of life better is perhaps to potentiate the role of beta genes. How can impact effectively? Through redox regulation, through nutrition, hormetic nutrition. So hormesis is going to be a very important word. And this is another aspect of my research where I have dedicated my present enthusiasm to characterize with my friend Edward Calabrese. He is in Harvard and he is the father of hormesis. And hormesis is a dose response biphasic dose response curve, where at low dose I have protection, high dose I have toxicity. This is very important in toxicology. This is the case of learning enhancing drugs, where you need to understand in a very well way the therapeutic range, which is for a biphasic profile. And NRF2, now, it's hormesis when we know persistent NRF2 good, detrimental long-acting activation of NRF2, maybe in cancer this has relevance, and this is hormesis. Just to make short a long story, I've dedicated many publications to hormesis, also to understand the yin and the yang of the traditional Chinese medicine in functional performance or neural system networks, and so putting the role of phytochemicals at the center of the relationship between cellular stress response and neuroprotection. These are other my recent last year contribution. That's why I try to understand how I can regulate cellular stress response as a means to obtain neuroprotection. I have used mushrooms, and with mushrooms we have been able to demonstrate that they are able to activate NRF2 because, as we will see after, they have polyphenols and activate protective system. Eriicium prevents the ethyl phthalate-induced apoptosis and mitochondrial dysfunction in neuroblastoma cells. And now come to our present situation. This is the background that has brought me to have a friendship and high stimulating research collaboration with Professor Rachan. And we have decided to go together and understand the possible importance of boswellia in our system. So, model of neurodegeneration from bench to clinic. And we have been working actively in these last months, thanks to Luei. We have been able to demonstrate in a model of brain damage, chronic inflammation-dependent brain damage, like traumatic brain injury, which we have characterized since 10 years, the role of boswellia. We have taken an extract from boswellia. We have characterized time dependence, dose dependence. We have found that 500 milligrams per kilo, which is very consistent with the current application in clinic, because rats 500 is 10 times less sensitive than humans. So you go 50 grams per kilo by 70, it's allowed between 2 grams per day. Can go to 4 grams, but we know that it's not toxic. So we can go up from 2 to 4 grams per day, like in some trials that we have left. And we have demonstrated how neurobehavior is very significantly impacted by boswellia extract. Let's call BS7. It's an extract that it's holy, magic extract. Why? Because, as you can see here, we have used the plasmids test and open field to explore the anxiety sign of brain damage or the locomotor activity, respectively. And we have a significant protection, as you can see here, brain trauma. After 30 days of treatment, brain trauma, 30 days neuroinflammation, and we treat with boswellia extract. Then we have PBI and significant protection. This is the open arm, open field, time in the center, time in the cross, so all locomotor activity, it's really significantly ameliorated after treatment with Boswellia. And then this is the histological results where we can see a vacuolar degeneration, really a massive cell death, and the protection with 500 milligram per kilo as measured by damage score here, TBI, after Boswellia extract. Then we have measured in the brain superoxide dismutase, glutathione peroxidase, malonaldehyde, and then we can see how after trauma and protective role of BS7 here, glutathione peroxidase, and then activity, MDA level, protection against lipid peroxidation. And this is consistent with a decreased synthesis of pro-inflammatory cytokines such as TNF-alpha, interleukin-6, and interleukin-beta. Together with this, we have also measured autophagy. And this is another very, very important chapter because we know that P62 can be moved away from the autophagosome and then activate NRF2, but in a persistent way. So this is something that needs a proper attention in future experiments. But so far we see that we have, again, autophagy. Autophagy, it's like inflammation. We might have reparation when it's short lasting, but we have chronic inflammation, which is degeneration. It's inflammation. And autophagy, it's the same, hormesis. But we cannot say nothing if we don't measure, but we know what to measure to dissect these two. So, little but with big significance, biological significance difference. And here, specifically, we have seen how the marker of autophagy, basically LC3, and also we have P62, they are increased, and the protective action of PS7. It's really, I would say, for me, it's amazing that we can really have a reason to go further. And then we have apoptosis, BAX BCL2, increased protection, decreased upregulation. This is my effort to be present here with results that are in a phase of we are completing this with NRF2, NFKB. In the next weeks we will have these results and we are ready to finalize this part of the study. And going with the same model of brain trauma, this is a mirror of what we will see also, hopefully, with Boswellia. Here, again, with mushrooms, we can see how Coriolis, Erysium, and the combination of Coriolis, Erysium, they give a very important protection. You see how many neurons we have lost, and with both Erysium and Coriolis better than in comparison with untreated. And then we have measured the marker of neuroinflammation. We can see after TBI and the protection. This is also true for NFKB. And here we can see the relatively the histogram for Western blot analysis for neuroinflammation at level of reactive astroglia and microglia. In all this situation, we have an evident protection by Erysium and Coriolis. And this is associated with induction of NRF2 and all gene controlled by NRF2, himoxygenase, shock protein 70, gamma glutamine systemic synthase, thioredoxin. In all this situation, the pacemaker is an RF2 transcriptional factor, which is activated in a favorable way with a biological impact by erythema Coriolis and the combination. Here we can see it's a signal of stress, but here it's a signal of reparation or mesis. And the way we have these results is connected to the possibility that we have measured in mushroom. I've been the first to demonstrate the presence in mushroom of polyphenols. Simply this. We have provided a clue to explain. And that's what we want to do also for our situation. Here we can see both LCMS and GCMS analysis showing the presence in Coriolis versicolor, but also in erysium of a number of compounds, especially polyphenols. We have kenferol, fumaric acid, and all other compounds that can have biological relevance. We have also used a model of Parkinson-induced animal model of Parkinson, where we have induced Parkinson with rotenone, and we have measured the effect of Coriolis. So, in some way here, these are polyphenols, but I'm missing the table of measured polyphenols in this presentation, but in the paper it's these are polyphenols. Okay. Routine here, ergosterol here. They are reported here just to be consistent. Ergosterol, catechin, epicatechin, apigenic, quercetin, kenferol, vanillic acid, ferulic acid, fumaric acid. They have been measured in mushrooms. Okay. So coming to from bench to clinic. Now we have seen that we can explore this redoxomic world and the effect of aromatic nutrients such as boswellic acid in vitro cell. Actually, we have model for blood-brain barrier, in here blood barrier, and also animal model of neurodegeneration like Alzheimer, Parkinson, glioblastoma, and to the clinic. As an example of, I'm the head of School of Clinical Pathology, so I'm at the center in the polyclinic of all kind of, with friendship and cooperative strength, with neurology. I'm a neurologist, also the first biochemist, but doctor and neurologist. But all my life, biochemistry. That's why I'm a clinical biochemist. Simple. So, gas chromatography since the first year of my studies, morning until night. So, so I can investigate Alzheimer, Parkinson, autism, multiple sclerosis. And this is an example of a clinical intervention, mania disease. I have launched this idea that mania disease is a neurodegenerative disease because we have symptoms that are connected with degeneration of inner-ear ganglion, cochlear vestibular ganglion. They are neurons. But I can measure through measurement of markers of tinnitus, vertigo, and inflammation. In fact, I have first characterized 10 years ago mania disease as an oxidant systemic disease that reflects the local inflammation, and I've provided antioxidant hormetic approach. Here we can see that by lipidomics in Meniere disease, we can measure the pro-inflammatory, anti-inflammatory steps, which it's very important, because from urine that I can receive from London, from Sweden, from everywhere, I can say if the subject before or after therapy or lack of nutrition change is redox state, pro-inflammatory, anti-inflammatory. It's not an easy matter. And in fact, in this model of neurodegeneration, Meniere disease, besides clinical sign of improvement after Coriolis treatment, after 6 months of Coriolis treatment, so these people, they get better mood and cognitive score, but also we have measured in lymphocytes NRF2 hemoxygenase, and we can see how Coriolis results in a significant protection, in a significant upregulation of NRF2, but also hemoxygenase, its shock protein 17, gamma glutamyl staining synthase, and thyroidox. We have the panel, so we will do the same, hopefully, with our products. And this together with marker of apoptosis. So we have BAX and BCL2. BAX is upregulated in Meniere disease and the protection by Coriolis, 3 months and 6 months, and BCL2 vice versa. It's quite interesting. And also looking at C-twin, we have upregulation of C-twin after 3 months, even more after 6 months. Glutathione is another marker of redox state in lymphocytes and in plasma, and we see how glutathione in Meniere disease is dropped 50% from 16 micromolar to 7.4, and the protection by Coriolis, 3 months, 6 months. GSSG, the opposite, and the ratio, which is very important. Again, is an oxidant disorders, the disease, so we have protein carbonyl going up in the lymphocytes of Meniere disease, and protection by Coriolis, 6 months, 3 months. And together with protein carbonylation, we have measured hydroquinones. Again, it's consistent. And luminescence marker of oxidative stress in the protection by Meniere disease treatment with Coriolis. We have investigated mitochondrial bioenergetic, mitochondria. So we have, because oxidative stress is also connected with the impairment of electron flows, especially complex 1, goes on oxygen, makes superoxide. And then we have complex 1, like in Parkinson, in Meniere disease, and the protection. This is very important because also it's a model of degenerative disease and the model of hormetic nutrition approach, which can be exported to many other clinical important situations. And complex 1, 2, 3, and complex 4. See? Complex 4 takes oxygen and produces water. If complex 1 works less, it can be an antioxidant molecule considered. If it takes less oxygen because works less better, then there's more oxygen for electron to make superoxide. So it's really something important. And then lipoxina 4. Lipoxina 4 is the protein which is crucial for all inflammatory pathway. If I manage to upregulate lipoxina 4, all other eicosanoids are downregulated. It's really the critical switch. In fact, we can see that we upregulate with hormetic approach lipoxina 4. This is in the plasma and in urine per milligram of creatinine. So here is the profile of eicosanoid, anti-inflammatory and pro-inflammatory that we have measured in Meniere disease and after treatment. So we can see we have a shift from pro-inflammation to anti-inflammation. And this is really relevant. Consider all the other marks. And consider the clinical endpoint that we measure. So at the end, no matter of biochemistry, we need to see if the patients get better or not. That's the end of the story. And we have a chance to correlate this. So the model is very important. It's a very important table where we can put our flower of interest. Pro-inflammatory, anti-inflammatory, all panel we have been able to, and we are able to measure. And that's why I'm at the end of my presentation, we have also in our lab animal model of autism. We study autism in C. elegans, cellular model, as we can, starting with autistic children, where we have designed possible treatment. The reason we will see soon. This is a publication. I have a small space at Johns Hopkins University where I'm developing mini organs biotechnology. And together we have produced a publication that last year had all general resonance in all the United States. And this because it has been shown that sulforaphane treatment stops autism. If I treat children with sulforaphane, they have no sign of autism. The typical pattern of autistic, they are repetitive, they get better. And every mother knows that the children get better when they are with fever. Fever, it's shock signal. So they start to understand maybe this is the way that we can impact what we have not been able to do so far. In fact, sulforaphane abolishes the pattern of autism and reduces symptoms. It's tremendous evidence why we don't have to try with our key. And so through metabolomics and lipidomics, we have been investigated a new dimension, which is brain organoid. We developed from a cell of patients from urine the brain. We are able to do this. And from pluripotent stem cells in current our lab, we are able to develop this. It's a mini brain in my lab where we can measure anything we want. Another aspect of my interest in my lab is this, just to finish, is flow-mediated vasodilatation. Working with polyphenols, it has been discovered that polyphenols, they modulate the vasodilatation of microcapillaries. That's why they work, because when we have a cognitive impact effect by nutrients, vasodilatation, everything gets better in my brain and all the neuroscience that is below. To convince you, yeah, this. Just, I want to do this video. It's a few seconds to convince you what happens. Looks like rain that becomes thunderstorm in terms of circulation. And we do this. I have a lab also at Shibara Institute in Tokyo. I've developed a collaboration between the two universities, and we can do this. So it's fantastic. You cannot say nothing if you don't measure. And that's why our life is in color, thanks to God, inshallah. And this is my idea, where I'm putting all my love, all my enthusiasm. Human donors, mini organ, not only mini brain, but mini skin. We are trying in Heidelberg to do mini kidney with Verena Peters. And the general idea from United States government is to develop a human on a chip, where I'm sorry for some pharmacologists, maybe we don't need any more animals. Okay? And thank you for your attention. Thank you very much.
Yes, thank you, doctor, for a very good presentation covering a wide range of things from brain injury to autism. Now we can take 2 questions. Any questions?
Hello. Thanks for this inspiring lecture. I wondered, though, you used a lot of these fungal extracts and named some of the very common flavonoids. So the question is, why did you, maybe you did try the flavonoids by themselves to have a more clear picture about what's happening?
Yeah, I come from a story where I used curcumin. I've been the first to demonstrate ferric acid in neurons effect. So all my life has been dealt with polyphenols alone. But Mother Nature gives a very important lecture. Don't forget. Harmony. The maniac will to dissect, in medicine, I mean, not in chemistry, in medicine, it's not a reward thing. You need the synergy.
Yeah.
I give you an example, which in German it's very hydroxytyrosol. I have a patent with Berkeley, olivenol hydrox, with a friend in Berkeley. This comes from, just coming to your answer,
Yeah, I mean, of course, every Galenics people know that. Of course, no. I was going for the scientific understanding, where you. I mean, I worked in 35 years in natural products, so of course, I always see these claims of additive, which are mostly there, and synergistic effects, which are mostly not there. They are often claims without any causal scientific background. Of course, you have these effects. We have seen that also, that you have triterpenoids which help you in the delivery of flavonoids by complexation and so on. Yeah, for bioavailability, that's an issue. For the activity itself, it's often less so. So in cell models, I'm a big friend of using really pure or compounds or pure mixtures also, where you have defined mixtures. Of course, in the real life, you're totally right. When you have all the intestine system, you have lots of secondary and transport and association effects in nature. Yes. That's what was the second part of my question, which is if you see any advantages of the versicolor extract versus the pure, what was it, routine or yeah. Because the flavonoids have intrinsically bad bioavailability. That's a problem, of course, in most of them.
That's a good question, because we see a significant difference. So the pure that we have done this experiment, if you consider like mushroom as an example, mushroom contain not only polyphenol, anthocyanin, I mean this kind of oligomer, which may have more activity than even the complex mixture of single polyphenols. So again, science, it's important to have purity. And this is the first, to understand. But at the end, to evaluate the biological endpoint, the clinical relevance, you have to consider a certain equilibrium as a matter of consistency. At least this is my experience. Thank you.
We can take this last question.
Hi, good morning. I'm Dr. Mohammed Al Marhoon, Senior Consultant Urological Surgeon, and I do treat a lot of male infertility. In male infertility, we have a big group of patients who are characterized as idiopathic male infertility. That means there is no known cause. However, the new research and science have shown that DNA fragmentation is a significant thing. And it is usually caused by oxygen radical oxidation and all of this. So treating these patients with antioxidants is believed to make some changes. So my question is, I have seen that you have mentioned a lot of antioxidants. If you are referring that Boswellia can have a role in this, I think we have to really look into this and make it happen.
Because I just received 1 week ago a paper to review pointing on the role of endocrine disrupting agents that we have in the environment, which make also through oxidation not only infertility but transgender transition. Understand? I mean, transgender. It's something that even the medicine is not ready, because if you are in the emergency and a transgender comes, you don't know before put something in the vein, how is his equilibrium in terms of coagulation, hormones, and it's a mess. You need to know. So, given that when you have this pollution, we are oxidant, they make oxidative stress, and infertility is one consequence. That's why Boswellia can be one of the rational approach.
So since Professor Rashan has not heard the question, I'm just stimulating that we should go in the direction of antioxidants from Boswellia for male infertility. Especially there is a high DNA fragmentations in these patients. Thank you.
Thank you.
Thank you.
If that doesn't work, I do have another drive as well. I get paranoid and put it on 2 drives.
bacterial strains. Since 1929, when Fleming first isolated and characterised penicillin, we have had a reliance on looking for new antibiotics from bacteria and from fungi, and we've been lulled into a bit of a sense of security that we'll always have new antibiotics, because we have, through the Golden Age, literally developed hundreds of very good antibiotics during that period. However, more recently, we've really exhausted the supply of bacteria and fungi that we need to look at to be able to develop new antibiotics, and more recent antibiotic development has come through chemistry, modifying chemical scaffolds to create different types of molecules with enhanced effects. Unfortunately, bacteria are a little bit better at this game than we are, and they keep developing ways of overcoming the action of those antibiotics. So we're constantly needing to develop new antibiotics. Let me try my other one in the meantime. I've sent it via email, yeah, but it's also on this one. Okay, so hopefully we'll get that sorted out in a minute. So basically my research attempts to look for, yes, the frankincense one, hopefully that'll work. To look for new antibiotics from plants. Fantastic, we're off and running. I'll skip through the first few slides to catch up to where we are. Now there are some bacteria that are considered highly resistant or totally resistant. There's at least 3 bacteria I know of that there is no treatment for at all. Klebsiella pneumonia, a Neisseria gonorrhea, and now a strain of, more importantly, of Mycobacterium tuberculosis. So we really do need to develop new antibiotic therapies very quickly to get us back on target. Unfortunately, at the same time, there's been a decrease in the development of antibiotics across this period because we're running out of new microbes to test, and instead we're having to go down the chemical synthesis. So in the past, it was all natural products. Now we're modifying the scaffold. We've been modifying the scaffolding for quite a while now to come up with new antibiotics. The chemists don't need new scaffolds to be able to continue with this. One way of looking at this would be to look for new antibiotics from plants, because before the development of penicillin, we actually used plant-based medicines as antibiotics. And many of you may know there's a lot of different plant molecules with very good antibacterial activity. Berberine is one that you probably know of. These are actually quite good antibiotics, or reasonable antibiotics. But you probably also know that there are literally hundreds, if not thousands, of papers published yearly on antibacterial activity of plant extracts. But there's very few plant molecules that have made it through to clinical usage, and this is a bit of an anomaly. Plant extracts themselves are great, but once you start separating out the pure components, you tend to lose activity. And one of the reasons for this is that plant extracts not only contain what we think of as the bioactive molecule, but they contain potentiators as well that allow them to work at high levels. By separating these out, we tend to lose this effect. So recently, my work has not only involved looking at plant extracts for new antibiotic molecules, but looking for these potentiator molecules as well, because I feel this is probably the most important way forward. And there are drugs on the market already that do this. Augmentin, which you may have heard of or may have used, contains beta-lactam, amoxicillin, along with clavulanic acid, which has got a similar structure, but it is inherently inactive. It competes with the, so generally what happens is penicillin will bind to beta-lactamase and be broken down and inactivated. Clavulanic acid, in the presence of this, it will compete with it. It will bind irreversibly and therefore essentially allow the penicillin or the beta-lactam antibiotic to work again. Now, this is one of the best known categories of overcoming these resistant mechanisms by inactivating the enzymes, but probably the most important one is that bacteria develop these efflux pumps. So when the antibiotic comes into the cell, it's pumped straight back out and it's not able to be effective.
So a very good target is to target these efflux pumps and see if we can wipe them out, and therefore the activity of the antibiotic can actually occur again. Now that leads us to where we're going to start talking about terminalia, which is the terminalia, that's another one of my projects, about Boswellia sacra, which is the topic that we're going to be talking about today as the antibiotic. I will skip through this fairly quickly in the interest of saving time because we all know about Boswellia, that's why we're all here, and in fact you probably know it a lot better than I do. So it's Boswellia sacra that we're going to be looking at. Again, it's been used for a long time. It has quite a number of different uses. It can be used to treat skin diseases, wound healing, urinary tract infections, respiratory infections, gastrointestinal infections, and interestingly, many of these involve bacteria. So that's the reason that I started getting interested in frankincense as an antibiotic or a source of new antibiotics, because of its myriad of uses that do involve bacteria. Now again, I will go through this fairly quickly, but one thing I did want to point out, as you all know as well, that frankincense tends to be graded, with the lighter colour being classed as higher quality, and as it moves to the darker colour, it tends to be classed as progressively lower quality. Now this is important for what I'm going to be talking about today, because the classing of the quality here is not necessarily relevant for all different bioactivities that we'll be talking about, as you'll hopefully see in a minute. So this work was in conjunction with Professor Rashan, and he provided me with oleoresins from various different regions, from the plateau, valley region and the mountains, from different plants. And he provided me multiple different samples of each of different quality. So we tested these, we extracted them with methanol and with water to get different phytochemical profiles, and then we tested them for antibacterial activity. To do this, I used two tests. Initially, we used a straight distaffusion assay to give us an idea of susceptibility, just a very quick test to see if we're seeing any antibacterial activity, and then we quantified it using a liquid dilution assay, which uses the chemical iodo nitrotetrazolium salt, which lights up to red when there's living bacteria. So it's very easy to quantify. Now to test our extracts, we decided to test against two panels of bacteria to start with, and initially we tested against gastrointestinal pathogens. In this case, were more concentrated on the bottom of this figure. And I decided that this was an important area to look at, because we tend to think of diarrhea and gastrointestinal diseases as a minor inconvenience. For a couple of days, your body overcomes it and you get better again. But in actual fact, diarrhea is a major killer globally, and in fact it accounts for nearly 10% of childhood deaths. So it really is a very serious disease. As well as that, gastrointestinal system is a perfect place for bacteria to develop resistance and to exchange resistant genes. So we deemed that this was a worthy project, and we initially did, as I said, the distaffusion assay to see if susceptibility. And what you can maybe see from there is the methanol extracts in all cases were actually reasonably active, and there was a lack of activity for the water extracts. Here we have against Escherichia coli and Enterococcus coli. So we did get activity across the board for these extracts, but it was always the methanol generally being a lot better than the water extract, and the water extract often not having activity at all. Similarly, we tried it with Staphylococcus aureus, with Salmonella Newport, and with Shigella sonii, and in all cases, again, there was appreciable activity for the methanol, all of the methanol extracts. Okay, now this table, I'm not sure if you can actually read it, particularly if you're down the back, but I've colour-coded it here for you to hopefully make some results stand out. We measured the MIC of the different extracts against the different bacteria, and in these assays, an MIC, a minimum inhibitory concentration, the minimum concentration you need to totally inhibit the growth of the bacteria, anything below 1,000 micrograms per mil was classed as a noteworthy activity. And I've noted these ones in blue on this table, so as you can hopefully see, that across the board, the different frankincense cultivars and the different grades for them, there was a widespread of very good activities across the different extracts. The other thing I'd like to point out is the bacteria we used were actually quite antibiotic resistant bacteria. So for instance, this E. coli that we used, when you're looking at pure antibiotics, to be considered as susceptible, it needs to have an MIC below 1 microgram per mil. The only antibiotic that hit the E. coli at this level was ciprofloxacin. So this E. coli was resistant to all of these other antibiotics. So I guess I'm just saying that to point out that these bacteria were actually quite resistant bacteria, and yet the frankincense extracts, many of them were actually giving quite good activity against these. We also did the same with a different panel of bacteria associated with autoimmune inflammatory diseases. And it might not be as widely known in this audience, but basically autoimmune inflammatory diseases are caused by external pathogens, and they need to be in genetically susceptible people. When I'm talking about autoimmune diseases, we're talking about things such as rheumatoid arthritis, lupus, Lyme disease, and ankylosing spondylitis, also such things as multiple sclerosis as well. These are all autoimmune inflammatory diseases. Now, they're all caused by, the ones that we're studying here, we're looking at the triggers of rheumatoid arthritis, ankylosing spondylitis, and multiple sclerosis. This one is caused by Proteus mirabilis, Klebsiella pneumonia for ankylosing spondylitis, and multiple sclerosis can be triggered by both Pseudomonas aeruginosa and Acinetobacter bailey, several Acinetobacter species, because the people have cell surface antigens that are similar to the bacterial antigens. So again, we tried these firstly in the distaffusion assay, Proteus mirabilis and Klebsiella. Again, we saw a similar sort of trend. Generally, methanol extracts were a lot better than the aqueous extracts, but we got activity across the board. Likewise, with the bacterial triggers of multiple sclerosis, again, we got inhibition for Acinetobacter. It was lower activity, but inhibition nonetheless. For the Pseudomonas aeruginosa, we got a lot less activity. Now, anyone who's worked with Pseudomonas aeruginosa may well already know that this is a highly resistant bacteria. It's pretty hard to inhibit it with most antibiotics because it does tend to be quite antibiotic resistant, but it also forms biofilms as well. So we did get some here that were affecting Pseudomonas aeruginosa growth, but nowhere near as effective as against the other bacteria. Likewise, we then measured the MICs of these to get an idea of how effective these drugs were. And again, the colour coding here, we have these ones that are coloured are indicating noteworthy or in some cases very good activity, like I have one here that's just over 100 micrograms per mil. That's down in the potent, down towards the potent category. And again, these bacteria are resistant, not as necessarily as resistant, antibiotic resistant as the previous set, but again, we're testing against antibiotic resistant bacteria here. So before getting onto the combinational stuff, I just wanted to summarise what we found for the resins themselves. Both the methanol and the water extracts had some activity. The methanol extracts were generally a lot more active than the water extracts. They were particularly effective against some bacteria with MICs down to or even below 100 micrograms per mil, which is considered very good in these assays. And the other thing that was interesting was generally grades 2 and 3 resins were actually higher activity than grade 1. So what you class normally in most categories as the higher quality resin was not necessarily the best against antibacterial activity, particularly against these bacteria. So we need to be aware of that. The best is not necessarily the best for what we're looking at. Okay, so that was interesting, but we decided to take it and look at the combinations as well, because as I said, I think it's really important to look at the ability to potentiate the activity of existing antibiotics, So to do this, we used or worked out the sum of FICs. I don't know if any of you have worked this out before or done this before. Essentially, it is a mathematical way of looking at the MICs of the extract and an antibiotic in combination and comparing it to the extract or antibiotic alone, putting it into these formulas and working out an FIC index. Now, if we do that, that will give us the ability to determine the class of interaction that we see. So using these assays, anything with an FIC or sum of FIC below 0.5 is generally considered synergistic, and that corresponds to about a 4-fold increase in activity minimum. We also have additive effects, which are also beneficial, but they are between the 0.5 up to 1 in the FIC. Non-interactive is generally between or greater than 1 up to 4, and antagonism is defined as anything above that. Now again, you may not be able to see these from a distance, but again, I've colour-coded them for you. These lighter coloured green ones indicate synergy. These darker colour are meant to be blue, but they're not showing up clearly as that. They are additive effects. The ones that are red are indicating antagonistic effects. And what you can hopefully see is that most of the combinations are unmarked here. They are non-interactive. They're combinations that are not helping, but they're not hindering the activity either. But importantly, we are seeing some synergy and we are seeing some additive effects, so the combinations are helping the antibiotics in their job. Where we found synergy, we then worked out the ratios, or looked at the ratios of these, not just the 50-50 mix, and used isobologram analysis to see where the ideal ratios were. Now for this one, chloramphenicol with the extract against S-Newport, you might notice that all of the results are aligning with the y-axis. That means that the antibiotic, which is this axis, is more responsible for the activity, and the extract is more responsible for potentiating that activity. In the case of the next one, ciprofloxacin and S. aureus, it's more of a mixed effect, which means that the one that's in the highest concentration is the one that's actually more responsible for the activity there. Now, there's a number of ways that this could be occurring. There's synergy, but the major way that we look for is through efflux pump inhibition molecules. Okay, they're probably blocking the efflux of these molecules out of the cell and allowing them to work. I will go through these other results a lot more quickly. We did this with K pneumonia and A. baileyii as well, and again, we got more, I guess, competitive sort of isobolograms here, indicating that both compounds are useful and they're both competing with each other for the activity. When we're looking at tetracycline, and the last one was with tetracycline, it was an important one because the resistance mechanism for tetracycline, the bacterial resistance, is almost always by efflux pump. You can get target modification of the ribosome as well, but the vast majority of the time is through efflux pump. So when we see potentiation of tetracycline activity, it probably is through efflux pump activity. Again, I'm going to go through these very quickly. We used the SALI in these assays, and again we saw a number of different synergistic and a number of different additive effects as well. And again, we performed isobologram analysis on those to find out the ideal concentrations. And I should have mentioned before that anything under the 1 but up to the 0.5 line is considered additive. Anything in this region is considered synergistic. So we only looked at the additive and the synergistic activities here. Likewise with SALI extracts against A. baileyii in combination with tetracycline, we again saw the tetracycline being the important compound of the mixture for the activity, so the extract was potentiating that activity. And again with the Hujari extract, same sort of thing was seen, and again we did the isobologram analysis against tetracycline and saw a similar sort of result. Okay, so obviously an extract or a combination is only useful if it's non-toxic as well. So we tested the toxicity of these as well in a number of different assays. We used a Artemia Norplay assay because it's very quick, and it is actually quite similar to the results you get from a cell line assay. We also used a cell line assay as well, using human dermal fibroblasts to determine the toxicity as well. Toxicity results are given here in the cell line assay. We looked at 50%, anything with 50% or greater mortality at 1,000 micrograms per mil was classed as toxic, and there was no toxicity measured in this assay. For the brine shrimp assay, anything under 1,000 microgram per mil as an LC50 is also classed as toxic. So again, no toxicity again in this assay. By working out the LC50s, it enabled us to actually report therapeutic indexes as well, which is simply a measure of how safe these things are compared to how effective they are. And generally, we consider anything with a therapeutic index over 4 as being quite safe. And as you can see, some of these are very high therapeutic indexes, meaning that, or indicating that they are quite safe. I will rip through the chemistry fairly quickly. We did also look at the phytochemical composition of these extracts as well. In this study, I focused on the terpenoids, mono, sesqui, and diterpenoids, mainly because we're looking at antibacterial activity here, and these are molecules that are known to have antibacterial activity. You will have heard yesterday a lot about boswellic acid and its derivatives. Here, it's the terpenoids that I'm more interested in, the volatile terpenoids. Now, there were quite a number of terpenoids that we found that were common across these different extracts, but I want to draw your attention to the one here that I've highlighted in red, and that's alpha-pinene, because it was the main compound across all of these extracts, no matter what grade they were, but interestingly, it also increased in the extracts that had the higher activity. Now, this is actually an interesting result because alpha-pinene has a number of antibacterial activities we know about. It can disrupt cell membrane, inhibit oxygen uptake, reduce ATP production, and these will all directly kill bacteria. As well as that, they will combine to bacterial proteins, which can not only necessarily kill those bacteria, but it could also block things like efflux pumps and therefore allow potentiation to occur as well. Now, in summary then, what we found that the Boswellia sacra extracts themselves were actually quite active, some of them against the bacteria that we tested, but probably more importantly, they were actually good potentiators of the conventional antibiotics, which means that we're able to use them to repurpose those antibiotics even against bacteria that are resistant to them. It was particularly good against tetracycline, which means that it probably is an efflux pump inhibitor, although that really needs still to be tested further. Okay, I'd like to finish up by thanking my collaborators and acknowledge my collaborators in this project. Obviously, someone who needs no introduction, Professor Rashan and Parag Das from Dauphin University. I had 2 students come to me from France to do a bit of an internship with me, Nicholas Faven and Manon Hawlett, that did much of the antibacterial testing that I've reported here. And as well as that, at my own university, Griffith University, Alan White, who was a chemist who did most of the chemistry for me. So, thank you.
Yes, thank you, Professor, for a very informative presentation. Now we can take 2 questions. Yes.
Thank you, Professor, for your nice and informative presentation.
Thank you.
I have a question regarding you said you tested a water extract.
Yes.
Have you tested the oil itself?
No, I haven't.
You will get better results for that.
Yes, I realise that I will in one of the assays, and the other one I won't. The distaffusion assay on an agar media, oils don't travel well through agar, so that's not a good assay for oils. The liquid dilution assay, though, on the other hand, would be a good one for that. I'm just more focused on the extracts, but you're right, the oils would be good to test. and the liquid dilution assay would be good.
And I
to
think
test.
you will get a better result than that.
Yeah.
Thanks.
Yeah, that's quite possible, yeah.
Thank you very much for an interesting lecture. E. coli is a very common organism for urinary tract infections. So
Yes.
my question is, have you tried in clinical practice using frankincense to treat E. coli? And if so, what is the dose and for how long?
No,
And do you trust it alone without antibiotics?
Right. No, I haven't actually done that. We don't have the ability to do clinical trials as such at my university. But you're right, that would be very interesting. Not only E. coli, some of the other bacteria that I looked at, Proteus mirabilis is also a very, very, very common urinary tract infection organism as well. And we had good activity against that too. As to whether it works clinically, I don't know. That's something I'd like to get at, but as I said, at Griffith University, we don't necessarily have the ability to do
So
that.
it seems frankincense is really touching me in my specialty from all the sides, urology.
Okay, yes, it definitely is hitting many of the bacteria and so forth involved.
Yeah, this is another thing we can work on.
Definitely,
Looking
definitely.
at frankincense and for urinary tract infection.
Definitely.
Yeah.
Well, one of the good things about conferences like this is to actually build collaborations, and maybe we can talk about that, looking at the clinical.
Thank you.
Thank you very much for your presentation.
Thank you.
It was informative. I have few questions related to the frankincense. Since in the frankincense, which you have shown that there are so many aromatic compounds, right? If we are concentrating on the antimicrobial activity, right? So why we are not concentrating on the only that aromatic compound which will affect the antimicrobial compound, which we can also synthesize by the retrosynthetic analysis with the organic chemistry. So every time, each in every part of the world, there will be not the frankincense tree. So if we will have that one, can we have the same result or not?
Can we have which?
Same result, same type of the result. And
With the different classes of compounds, yeah. Well, you're right. Frankincense is a very complex mixture of molecules. As I said, I chose to concentrate on the terpenoids because they're known to have antibacterial. You're also right. A lot of other polyphenolics, like your flavonoids and so forth, have a similar sort of activities as well. The terpenoids, though, are of interest to me because they're all known to have activities like this, but many of them don't only have antibacterial activities in their own right. Pinene, for instance, as I was saying, it can actually also synergise other things. So I like to find molecules that are antibacterial in their own right, but I even get more excited when I find the synergisers because this gives us the ability to look at drugs that have been very effective, like penicillin, and are now no longer as effective, to actually synergise them and get them effective again. So I guess I targeted those molecules largely because of that, but also some of these smaller terpenoids as well. They have very fundamental roles. A lot of them affect membrane fluidity and so forth. And interestingly, and correct me if I'm wrong, but I've never come across a terpenoid that bacteria have developed resistance to. So to me, that's a very important factor as well. Hopefully that answers.
just I want to continue. If we'll have time or not? Last question, actually. What I think, because same type of the compound we can synthesize with the so many amine, right, which will also show the antimicrobial activity. So why should we cannot concentrate on there and we should concentrate here? So because it's very difficult to extract first and then go to the retrosynthetic analysis, right? So there are so many amine compounds are available, which is very highly toxic, you can say, or antimicrobial compound. So why we are concentrating here?
Well, that is a good question and it's a good point. You're right. A lot of these compounds, particularly the terpenoids, are available in a lot of other plants. You can make them as well. But it's not just the terpenoid. As I said earlier on in the talk, that there is a lot of very good extracts with activity that, when you see the amount of plant-based drugs that have actually made the market or clinical usage, it's actually surprising that very few have, and that's because when you get the pure molecule, a lot of the time you lose the activity. It decreases a lot. So in this case, in most cases of pharmacology and so forth, it's better to have a pure compound, and I agree that is generally the best option. However, in antimicrobial activity from plant-based molecules, it's often better to have a mixture because you've got different molecules synergising each other. So to say that alpha-pinene has these activities, that's all well and good, but other molecules in there help it to have those activities. So yeah, you're right, it is a good idea to have the pure molecules, and why go for the more difficult source of getting it, but it's not necessarily just a single molecule that's doing it.
Thank you. I will try to make a short question. Thank you for your inspiring talk. And you mentioned that some of your bacteria that you tested, for example, P. aeruginosa, forming biofilms. Have you done any studies to actually see if some of your compounds actually inhibiting biofilm formation, and that's why the other components are more active in these mixtures?
Yeah, also a very good question. You're right. The biofilms is a very important part of that. And yes, we have done some rudimentary experiments on that. It's not advanced enough for me to present it here, but it does look like there is some effect on the biofilms as well.
Yeah, I have two questions. First of all, of course, we don't see antibacterial resistance usually when there is a physicochemical effect. So all these membrane-active antibiotics are anti-resistant type.
Absolutely.
The problem with them is very often that you need either, like in the terpenates, concentrations which are not good enough for treatment or will give really severe problems to the system.
Yep.
Or also the other membrane-active ones have more severe side effects when the specific antibiotics which target some metabolic process. And in plants, I totally agree with you. I mean, plants usually do not produce a single very active compound. They produce a plethora of, let's say, different flavonoids as antifungals, for instance, or antibacterials. And each of them individually in that concentration would not be effective.
Yes.
But what we often forget is we want to use it for treatments. Plants use it for prevention. And that's a totally different situation, because many plants, once they are really infected, they die. Because the concentrations, the additive, mostly it's additive, sometimes also synergistic effects, but mostly additive, they are not strong enough to defend a heavy infection.
Yep.
And thus, it's, I think, very often a misconception we have that the plant, what is working in the plant, will be working for us as humans equally well. It does only rarely. So that's why we so rarely find good antibiotics from plants, better from fungi and so on. And the reason is that with this low concentration which you have there, that is preventive concentration if single bugs come there and try something and then they are inhibited. But it's not good enough if you already have this strong infection somewhere, or you would not be able to get enough. For instance, flavonoids have a very bad bioavailability profile. And then it would be very, very hard. It's terrible to get enough into your system to fight an existing. So that's the difference between plants, which we often forget. And
terpenoids, the bioavailability is even worse. But again, it comes down to how you're treating or what you're treating. For instance, topical treatment through skin.
Topical treatment is different,
even
of course.
Gut treatment as well, where you don't want to absorb the compounds into the necessarily into the bloodstream.
Or inhalation,
Yeah. So things like that are actually suited for this, but you're absolutely right. There is the additive effect from this, and for plants it is preventative as well. One of the things that I would point out, though, as well, is one of the panel of bacteria that I looked at were the autoimmune inflammatory inducing bacteria, things like multiple sclerosis, rheumatoid arthritis, and these are preventative as well. I definitely do not advocate giving someone with rheumatoid arthritis an antibiotic, a pure antibiotic, every day for the rest of their life. All you will do is develop resistant strains. What is more sensible is preventing it with something that is a crude mixture, or a relatively crude mixture. Because as you said, there's a number of different antimicrobial molecules that will actually look at it. and there's a number of synergisers there which will allow you to actually minimise the amount that you need to treat. So in that respect, the autoimmune bugs are actually looking at the same sort of thing, looking at blocking rather than inhibiting rather than treating.
I'm not an expert, but I'm guessing that once you have developed this autoimmune problem, killing the bacteria afterwards doesn't help you with your autoimmune problem anymore because it's already there from the cross-reactivity.
Yes
is that
and
right?
no.
Or is it even helpful?
Yes and no. I'll take multiple sclerosis as an example. Now, people that have or are prone to multiple sclerosis have the genetic makeup. They've got antigens on their cell surface that are very similar to antigens on these bacteria. They get infected with the bacteria. Their immune system makes antibodies against that, which it absolutely should, to get rid of the bacteria. Unfortunately, they also attack the myelin sheaths on the nerves and destroy those. When the bacteria is gone, though, and the antibiotics go, the effects of the multiple sclerosis dissipate. They get infected with that bacteria again, and it comes back, and it's an additive effect. It gets worse and worse each time as well. So, yes, you do get better, and the bacteria is gone once you've got rid of that, but it does come back with repeated infections, and it actually accumulates.
Thank you.
Now we'll
That wasn't
yes. Now we move on to the next speaker.
Sorry,
Dr. Hisham could not make.
sorry.
So
I have questions.
Yes.
I have question. I thank you for your presentation. It's very interesting to mix with the natural ingredient for synergy and the natural ingredient with the pure compound for it. But no, forget, this is my question because I am interested by toxicological approaches. No forget that there will be toxic synergy between. And my question is how to report the toxicity shown on Artemia at the toxicity threshold in human for possible use as an antibiotic adjuvant ingredient for Boswellia ingredient. It's very important because if you use mixture with Boswellia and the pure compound, you can to change toxicity of each.
Absolutely. I didn't have time to go through that in this, but we have actually tested that with these, and the combinations themselves are still non-toxic as well. You have to be aware it's not only the plant-based molecules, the terpenoids that have toxicity, and terpenoids definitely have toxicity, but the antibiotics themselves are toxic as well. So by putting them together, you're right, you do have the possibility of increasing that toxicity as well. We have checked it though, and we've not detected an increase in toxicity in these assays.
Yeah. Thank you.
We haven't gone into clinical studies or...
Yes. Our next speaker is Dr. Mohammed Suleiman from Switzerland and Jordan, respectively. He will be making a presentation on treatment of GBM. Yes, welcome, doctor.
Okay, assalamu alaikum. We moved now from the lab to the clinic. My talk today concerns treatment of glioblastoma with high dose and long duration Boswellia sarca preparation, a case report. As we know, glioblastoma is the most common and aggressive primary brain tumor in the adults. The overall incidence of glioblastoma is a relatively low incidence rate of around 3 to 4 cases per 100,000. The exact causes of glioblastoma are still not fully understood. Genetics factors, environmental exposures, and certain predisposition conditions such as exposure to radiation or certain genetic syndromes may influence the risk of developing glioblastoma. Depending on their origin, glioblastomas can be divided into primary glioblastomas occurring de novo and has bad prognosis, and accounting for about 90% of all glioblastomas, and secondary glioblastomas which occur due malignant progression of the lower grade astrocytoma, which has better prognosis. Common clinical manifestations of glioma depends of the affected location inside the brain. Mostly of patients has headaches, seizures, cognitive changes, motor weakness, sensory changes, visual disturbances, speech and language difficulties, personality changes, nausea and vomiting. What is the standard therapy? The standard therapy is 3 modalities: maximal surgical resection, not always possible, depend on the location of the treatment, the health of a patient. More than half the patient, we can do just biopsy. Followed by radiotherapy plus concomitant and maintenance temozolomide. Despite maximum treatment, the cancer almost recur. The radiotherapy, the duration of the radiotherapy is 6 weeks, 5 times weekly. We deliver 60 as a curative dose, 60 gray in 30 fractions. We have recently added to the standard treatment, which surgery followed by radiotherapy, temozolomide. With 3 modalities, we have better progression-free survival. We move from 6.9 months versus 5 months, and overall survival from 14.6 versus 12.1 months. Despite recent therapeutic advances, glioblastomas still represent the most common and devastating primary malignant brain tumor, and the prognosis remain poor, with 1 and 5 years overall survival rates of 29% and 3% respectively. One common complication associated with glioblastoma is brain edema, which is accumulation of excess fluid in the brain tissue. It represents a major cause of morbidity and mortality in glioblastoma due to the high risk of brain herniation in up to 60% of patients. Cerebral edema influences the clinical course and prognosis of the disease. Brain edema can occur for several reasons in the text of glioblastoma. The right color represents the edema. And we show here the edema shift to the midline to the other side. The mainly type of edema in glioblastoma is the vasogenic edema. We know glioblastoma often disrupt the blood-brain barrier, which normally separates the brain tissue from circulating blood. This disruption can lead to leakage of fluid and proteins from blood vessels into the surrounding brain tissue, causing vasogenic edema. There are many molecules involved in the glioblastoma induced brain edema. The first one is the vascular endothelial growth factor. And we know in the glioblastoma, we have excessive production of vascular endothelial growth factor leads to formation of abnormal blood vessels that are excessively leaky and dysfunctional. And we know the vascular endothelial growth factor decreasing the expression of tight joint proteins. We know in the brain capillary endothelial cells are the tight junction protein are connected with this protein complex that fuse the endothelial cells. So we see here, this is the proteins. And when brain tumor develop particuloglioblastoma, tight junction become permeable due to the release of angiogenic factors and changes in protein component. These events cause blood-brain barrier breakdown and vasogenic edema. The second molecule is the aquaporin 4. Is a water channel protein that regulates the movement of water in the brain, and glioblastoma cell can upregulate the expression of aquaporin 4, which increase water influx in the and edema formation. The third one is matrix metal proteinase. We know can disrupt the integrity of blood-protein barrier, leading to increased vascular permeability and the accumulation of fluid in the brain. And we know also the glioblastoma release various cytokines and chemokines such as interleukins 168, tumor necrosis factor alpha, and transforming growth factor beta. And these molecules can promote inflammation and increase blood vessel permeability, contribute to the brain edema. And we have the hypoxia-inducible factor 1 and prostaglandin, which can, the glioblastoma, we know the glioblastoma can produce prostaglandins, which are hormone-like molecules that regulate various physiological processes, including inflammation. Prostaglandin can increase blood vessel permeability and contribute to the development of edema. Yeah, for the edema, the standard treatment is dexamethasone. And we use in our daily work dexamethasone pre-operation, after the operation, with their radiotherapy. Why we use dexamethasone? It works through several mechanisms: stabilization of blood-brain barrier, has dexamethasone anti-inflammatory effects, reduction of vascular permeability, and reduction of angiogenesis. As I told before, glioblastoma produce the angiogenesis, has this activity too. But dexamethasone, we know, is a very and excellent medication, but up to 50% of patients develop side effects, sometimes really severe side effects. The top 4 side effects: the Cushing appearance, hyperglycemia, psychotic symptoms, and gastrointestinal issues, such as gastritis, bleeding. Yeah, this is a very interesting study, was published 2021. And this study included 10 articles with a total of 2,230 glioblastoma patients. This meta-analysis showed or revealed that dexamethasone was significantly associated with the poor prognosis of glioblastoma patients. And dexamethasone can increase 48% death risk of glioblastoma patients. The conclusion of this interesting study, the results suggest that the use of dexamethasone was associated with shorter overall survival and progression-free survival of glioblastoma patients, especially for newly diagnosed patients or glioblastoma patients treated with dexamethasone over 2 milligram daily. This dose is very, very small. Usually we need to treat symptoms of brain symptom. We need at least 4, 6, 8, 16, 24 milligrams. This is a very low dose, but still with this dose, we will, in this study showed we have less overall survival with more than 2 milligram daily. And the conclusion recommend the development of new therapeutic drugs for edema is urgently needed. Why dexamethasone is associated with lower overall survival and progression-free survival? I found that maybe this could be the answer from the publication of Dubinsky. Through attenuating the innate and adaptive immune system, prolonged exposure to dexamethasone could inhibit immune effector response against the glioblastoma. Dexamethasone could induce leukocytosis and affect the tumor immune infiltration and survival glioblastoma patients. Leukocytosis, we have more immune cells, but the distribution is abnormal and the cells are abnormal. Now I move to my case. It is an 43-year-old university professor. He started showing trouble speaking, understanding words, reading and writing, and headache. The investigations done as routinely with brain MRI. This examination showed brain mass in the right parietal lobe with extensive edema. On April 2017, underwent a stereotactic biopsy. Was due to the deep location and definite histology. Due to the deep location, was not possible to do maximal surgery. The definite histology revealed glioblastoma with IDH1 mutation. Generally, it has better prognosis than widely type. Because today we divide glioblastoma in IDH1 mutation or widely type. Widely type has poor prognosis. He had the classical treatment, the 3 modality, radiotherapy, chemotherapy, and timodal, temozolomide. And I added Boswellia sacra. I started with 1,800 milligram daily. We divide three times daily. The total is 1,800 milligram. And his dose increased after 2 weeks to 4 gram till now. That means since 6 years. Dexamethasone 16 milligram day during the first week of the treatment, and those adjusted later to be at the end of the radiotherapy 1 gram twice daily. The result of the treatment, standard treatment plus Boswellia sacra, we get after 2 years, follow-up, the glioblastoma was disappeared completely. Steroid therapy could be tapered and stopped rapidly. Combined treatment was tolerated without any objective side effects, and the patient since 6 years in complete remission, and he joined his work at the university. He refused. I told him maybe we can reduce the Boswellia sacra dose. He told me I do regular every 3 months lab investigation, liver enzymes, kidney enzyme, CBC, all are normal. But he refused to change the dose of Boswellia sacra. He told me when I try one time, I have problem with the concentration. I feel me fatigue. I have fatigue. I can't concentrate it. And he told me, I am happy, I want to continue with this dose. This is the last Emery shaded, shade brain from 21/5 May 2023. This exam showed complete remission with a little bit edema. Why glioblastoma is useful? I found possible mechanisms of action. We know the Boswellia has the properties as anti-inflammatory, inhibition of the production of leukotrienes, has Boswellia, as we know, anti-angiogenic effect, antioxidant effects. The conclusion, the result really is promising with long survival. He has good quality. He usually we observe with patient after radiotherapy, chemotherapy, problem with the memory. He told me, I don't feel any difference. I don't know. It's that subjectively he didn't any problem with his job. Also, what is interesting, after 6 years, no gastrointestinal side effects with high dose. He take the Boswellia with meal. All. This is our recommendation. No gastrointestinal side effects with high dose and long duration treatment with Boswellia sacra. Our case suggests that Boswellia may serve as additional treatment to topical steroid treatment. I added also maybe we have anti-tumoral effect, not just anti-edema effect. The value of long duration with high-dose Boswellia sacra in treatment of patients with glioblastoma should therefore be further investigated in prospective therapeutic trials. I have more than one patient. I have 3, 2 years, 3 years, also glioblastoma, and they did till now very well. And no sign of recurrence, and they have good quality. This is what some patient did not well, but I see now in this, as we know, glioblastoma, nobody will be cured, no cure. Till now, when I see some patient with long duration survival, it's an advantage. And when I give Boswellia sacra, we don't have side effects. Why not? I have just a small number of cases, but I think when we have the opportunity to observe, maybe to do trial, clinical trial is not easy, but maybe when we collected more and more cases like this, maybe we have after 2, 3 years or 4 years more information, solid information about Boswellia sacra in treatment of brain edema, or the glioblastoma-induced brain edema, and we can maybe have more information about survival and quality of life. Before I want to close my speech, I want to thank my teacher, Dr. Rashan. He has had really, for his constant support, he always provide me with useful information about frankincense, and he showed me the phototherapy. Without Dr. Rashan, maybe I didn't did this. I have now my diploma in phytotherapy and complementary therapy. I did thanks Dr. Rashan. I am happy to do it, and thank you very much. Really, I thank you. He is my teacher.
Thank you, Professor, for the very informative talk. Now we can take 2-3 questions. Any questions? There should be some questions.
So very impressive and very stimulating also for future investigation. So my question, do you also have patients suffering from other tumors than brain tumors? Maybe breast or colon tumors or any other kind of entities?
With
yes, with Boswellia sacra.
with Boswellia, I treated regularly basal cell carcinoma, but with oil. The result is fantastic. Dr. Rashan, he told me I tried to add it Boswellia sacra as extract. I did, and the result really fantastic. In my daily job, in my clinic, I have many old patients. I treat with cancer. When I did the clinical examinations, sometimes I found basalioma or the epithelial cell carcinoma. I proposed to my patient to try with this with oil. After 5 or 6 patients, I found the result is really fantastic. Now I do it regularly. I give regularly Boswellia oil to treat basalioma and platinum carcinoma, squamous cell carcinoma, in English. Yeah, I try regularly also patient with Boswellia for ulcerated breast cancer with oil and capsule. The result is really more promising. I give for prostate without alone, without ADT, because ADT is contraindicated to give together.
When you say prostate cancer, is it also active against benign hyperplasia of the prostate? Have you any idea?
I have some patient with fibroadenoma and cystic diseases in the breast. I give some, and we get rapidly result against mastectomy. Against pain is fantastic. I have seen trial from Italy was published for fibroadenoma, and the result was good. For this reason, I take the result, and now I treat also my patient with Boswellia. Yeah.
So prostate hyperplasia is a huge problem in men above years of 60. So maybe this might also be a future application of frankincense.
Yeah, for mostly are young patients between 30-40 years, and the only treatment is anti-conceptual, anti-hormone therapy. And many women don't like to have this kind of treatment. For this reason, I think Boswellia could be a good alternative.
Thank you.
Concerning the prostate benign hyperplasia, which dosage you use?
Yeah.
Or prostate.
Mine
Cancer.
preferable dose usually 2 to 3 grams
per day.
per day. For brain disease, I go more, 4 to 5 a gram.
Yes, so one more question we'll take.
Thank you, Dr. Mohammed. It's very, very interesting. But your results are more than interesting, but their limit is the size of the cohort.
Yeah.
Yeah, and that you had used. In addition, I found that you increased to doses until 4 gram, 4,000 milligram. Benefit risk ratio is important to know if the positive effect is more important than negative effect. It's very, very important for the safety, safety of product. And I finish here. I think that this result must be published because this part of your result is scientific phytotherapy, because phytotherapy is not every time scientific. More time is based on ethnopharmacology in tradition. But this work is very scientific and clinical, important. I just want to indicate that in this topic we have be careful with safety of product and with rapport benefit and the risk. Sorry.
Concerning the dose, there are many studies were published about 20 years ago concerning doses. And this study, we saw the response is dose dependent. That means with the authors try to give 800, 100, 2,200, 1,800. And the result, every time when he increased the dose, the result was better. And he tried to 3 grams, 3,000. Well, the better compared with the low dose was better. For this reason, for the glioblastoma-induced edema is standard, was studied 4 grams. That is, but I told we have to observe the patient. But with my daily job, I never saw problem with liver enzymes. Sometimes the patient, after the first day or the intake, sometimes has slight nausea. In this time, what I have did against this side effect, I told the patient to stop the treatment, to wait a couple of days, and to retake. I had just, I want to say, success. And after I increase the dose slowly. That means when I start with usually 1.5 gram, I increased within 10 days or 2 weeks to reach 4 gram. As slowly I go up. Yeah.
Dr. Suleiman, you have mentioned a lot of interesting or stimulating things regarding prostate cancer, benign prostatic hyperplasia, and I may add also chronic prostatitis. All these 3 things are very common in urology, and I mean, they are touching a lot of population. So you have mentioned what I can see is anecdotal experiences, but we need really to prove this by controlled studies, including prostate cancer with ADT as a control in BPH, that is prostate hyperplasia. We need to look at sizes and give treatment and then monitor the sizes of the prostate. Remember that there are now a lot of new techniques to deal with the prostate. I just would like to stimulate you that there is a resume, which is steam therapy for prostate, which shrinks the prostate. It has made the surgery very quick and harmless. If we can bring something even more harmless and natural, that will be even better. Chronic prostatitis, with all our medications, we are still failing to make the patient satisfied because it's a chronic disease, demanding. So if you can put them on something natural for a long time and it will make effect, I think we need to prove this and publish, hopefully.
For prostatitis, chronic prostatitis, I suggest a kind of treatment. I do combination. You know, prostatitis, many patients have experience with different antibiotics. And I have just my personal experience. Boswellia with sodium bicarbonate. You have to try. Maybe we have success. That is first. I know we have to do trials, but not with phytotherapeutic medication or the high pflanzen, with phytotherapy. We don't get easily a sponsor. You know, phytotherapeutica is many factors are small and don't have huge resources, financial resources. For this reason, it's not easy. It's easy to say we do trial, but not easy. I think the first step now to collect cases, and when we have good result, we can provide the manufacturer. We have good result. Maybe we found sponsor. For this, my recommendation: we can start trials. It is difficult, but I suggest. I think, or I believe, that to collect the cases is the easiest and the fastest way to get good solid information.
Retrospective trials.
Yes. Before, it is not easy. I propose to do in our university in Bern trial with Boswellia. Nobody has accepted this. The idea has been refused. For this reason, I think we have to start step by step. A retrospective study, we collect cases, and when we show we have good result, maybe with the time, can we do trials?
Dr. Suleiman, in Oman, this will not be allowed unless we prove to them scientifically that it is effective, then only will be able to practice it. So
in the lab, we have. I think, you know better than me. We have enough information. We have to move to the next step.
We can do clinical trials. Let's collaborate.
But before we have to collect the country dependent. What I see in Europe, it's not easy. Not easy. Maybe in Oman easy.
Yeah.
I don't know.
Yeah, I think we
This
can
is
have
exactly
more discussion.
the idea I got on this discussion. When we would have a retrospective trial here, maybe we have a good reason to perform a prospective trial in Oman. And the situation in Germany and all the other European countries, the Western school medicine is rather reluctant against phytotherapy, whatever it is. Maybe there is a better chance in the Near East. Maybe we have better chances here. And as you said, it is not easy to find a sponsor for a clinical trial.
Yeah.
Clinical trial is very expensive. Maybe this can be done with a proposal to the government. If we would have governmental financed trials, then we are independent of enterprises, of companies, and this would be the way to lead in the new direction. Yeah. And Boswellia might be the right compound to start with.
I can just add
this is the land of frankincense. It should be easier.
I just would like to let you know that funding for research in Oman is not that difficult. We have the Research Council, we have the university with His Majesty grant and different grants, and sometimes you can even collaborate with companies if they find your idea is really strong.
So now we conclude this morning session with 3 wonderful presentations with very stimulating questions from the audience. And in particular, I like the discussion put forward by Dr. Madhun. Thank you very much. Now let's go for tea. And our next session will scheduled to start at 11:45 by Dr. Shaukat, one of my colleagues. Thank you very much.